Welcome to this briefing of the Jerusalem Press Club, JPC, another one in the series of webinars we have been running for the international press since the beginning of the COVID-19 crisis. I'm Uri Dromi, Director General of JPC, and today we host Professor Yaakov Nahmias, the founding director of the Alexander Grass Center for Bioengineering at the Hebrew University of Jerusalem, and a faculty member of the Center for Engineering and Medicine at Harvard Medical School. Professor Nahmias, thanks for taking the time to be with us today. So when the whole world is racing toward a vaccine or a new medicine to combat COVID-19, you come up with an existing medicine, the fenofibrate, sold under the brand name Tricor, that could potentially downgrade COVID-19 severity into nothing worse than a common cold. Can you tell us more about it?
Thank you, Uri, and thank you everybody for joining us today. There are a couple of important things to understand about viruses. Viruses are very simple packages of protein with a tiny bit of genetic material inside them. They can't make new viruses. They have to infect our cells. In the case of the new coronavirus, it has to infect lung cells, change the cells in such a way that the cells start producing more virus. Because of that, viruses are really sensitive if you understand what they're doing inside the cells. If you find out there is a pathway, there is something that they need to make more viruses and you can block it, then that's their Achilles heel. So over the past 3 months, we've been working together with Professor Benjamin Tanover from Mount Sinai Hospital, trying to understand what the virus is actually doing in human lung cells and trying to find a cure for it, essentially a way to stop it. So allow me to share a few slides that I'll go over to explain our major findings. So SARS-CoV-2 is the scientific name of the new coronavirus. This virus causes a disease that causes respiratory failure in patients that we call COVID-19. You can think about them as roughly the same thing. One of the first things that we see when you look at lung cells that are infected with SARS-CoV-2 is that a lot of things change, but one of the major things that change is all of the genetics that deals with lipid metabolism. So out of all of the genes that change in the human lung, 65% of them are metabolic, and actually 20% of them deal with lipid metabolism, essentially fat. This is something very strange. Now we see that not only in human lung cells, but also from biopsies taken from patients. We feel very, very confident this is indeed what we see in COVID-19. When you look down at it, it looks like everything in the cell has been rewired. Cells that are infected with SARS-CoV-2, with the new coronavirus, take in a lot more glucose, and you can see in the photo a lot more green in the cells. This is a tracer for glucose. And then they take this excess glucose and make a lot more fat. So this is a picture of normal lung cells, and here you see the human lung cells infected with the virus. And you can see all the green and red. The red is all the membranes that make new virus, and the green is all the lipids that accumulated inside. So there is fat in the lung, and it's not supposed to be there, okay? In the lung cells themselves. Why is this happening? So molecularly, it's happening because the virus really needs this fat. Well, it needs fatty acids to anchor itself to the cell. On spike protein needs this palmatic acid. It also needs cholesterol to make a lot of membranes. That's all the red that you saw a second ago. This is the factory inside the lungs that make new viruses. In order to get these two up, well, it blocks the cellular ability to burn fat. So this green arrow is essentially cells taking fatty acids and burning them to ketone bodies like acetone. This is how we burn fat. And this is controlled by one transcription factor, one genetic element called PPR alpha, and it's completely shut down, both in cells and in human biopsies. So this is interesting because the second we saw that, we knew we might have a drug, because fenofibrate, a drug that was approved by the FDA many years ago, activates the precise thing the virus needs to shut down everything else. And when you give human lung cells fenofibrate, you see this. So these are cells, again, all these red membranes where the virus is being made. When you treat them with fenofibrate, suddenly this red goes away, okay? This, over here, you can actually see the amount of virons produced by the cells infected with the virus when they're treated with fenofibrate. After 5 days, there is almost no virus left, okay? And this is log scale. So we're talking about 2 to 3 orders of magnitude. So times 1,000 times less, 10,000 times less virus than before. So a few points, and this is really the last slide, and open it up for questions. This is a study that both Ben and I financed from our own grants. This hasn't been any pharmaceutical company or commercial entity behind it. The work that we're doing here is fundamental. It's carried out on human lung cells. It's not a cancer. It's not a drug screen on cancer cells or on monkey cells. This is human lung cells with the full virus, with validation of a lot of these things from biopsies. We're starting animal work this week in Mount Sinai Hospital, and we hope that clinical studies are going to start in 3 to 6 weeks. Thank you very much.
So, thank you. It's amazing your ability to tell laymen about these complicated processes. I'm sure you're aware of some reservation raised about this breakthrough. For example, Professor Eitan Friedman of Sheba Hospital says that before coming to any conclusion, it's not enough to experiment in the lab, but you need to test the medicine on many people.
Right. So I wouldn't say that we came to a conclusion, but we have a very promising candidate. One of the biggest problems with clinical centers in Israel is that it takes quite a lot of political capital to start getting them to move toward clinical studies. I would say that I would just point out, for example, that we got an institutional review board approval to get retrospective data from patients that could validate our results in the beginning of April. We're in the middle of July, 2.5 months later, and we still didn't get this data. So we think that there needs to be significant push to get into a clinical style trials in Israel and abroad. And this is one of the reasons we are going to the media.
So what can you elaborate on the next steps? What you mentioned the fact that they start in New York tests on animals.
So that is right. So right now we're going into animal studies, and hopefully they will conclude in about 1 to 2 weeks. But regardless of the animal studies, we can go into early stage clinical studies. The interesting thing about fenofibrate is that it has a very good safety profile. Fenofibrate is the actual indication for it is dyslipidemia. It is given for patients that have high levels of triglycerides, essentially fat in their blood, so high cholesterol. Because of that, and because dyslipidemia, high cholesterol, high lipid levels is also a major risk factor for the development of COVID-19, the indications to use this drug are already there. That means clinicians can decide to prescribe this drug right now. We don't recommend that. We think that we need to go into clinical studies, but we hope that the approval of the clinical study is going to be a lot faster because we're not really changing the indication.
There's another reservation, or rather criticism, is that COVID-19 is a pandemic with PR. And sometimes scientists and pharmaceutical corporations are too quick to announce breakthroughs just to remind the world that they're in the race. You're not one of them, are you?
I hope not. You know, we spent the last 3 months of the quarantine working very, very hard to get to the point where we are breaking off the mechanism and understanding what the virus is doing. My biggest problem, right? The reason that we started this is because a lot of different groups around the world are using cancer cells and monkey cells to study this virus. These are excellent models if you're trying to understand how the virus gets into the cells or replicates. The problem is that you can't understand what the host does. You need to work with lung cells and you need to work with human cells, and very few people were doing that. Beyond that, very few people had the tools that we do. You know, understanding metabolism is something fundamental that my lab is doing, and we did it in hepatitis C, you know, over 5 years ago. So the fact that nobody was looking into that, you know, was the major driving force for us to push this as quickly as possible.
It sounds so simple, unbelievably simple. Like what you're saying to the world is, why rush to find a vaccine or a medicine when you have one already?
I wouldn't dare saying that. I think we need every single weapon in our arsenal to fight this pandemic. It has caused significant human suffering and massive commercial problems around the world. We need every single weapon in our arsenal. My biggest fear regarding vaccine development is that current studies suggest that antibodies for the new coronavirus would only stay in the blood for 3 to 6 months. That might not be even enough to protect somebody for the entire year. Then if this is true, if this pans out, then we're in a big problem with vaccines. So we need another line of therapy. Medicine like remdesivir is specifically targeting the viral proteins. And one of the biggest problems with antivirals that target the viral proteins is that viruses mutate, and they mutate extremely fast. That means that we might have an antiviral drug that works this season and not next season. On the other hand, if you're targeting a host pathway, like we do, we're targeting a human protein. Humans don't mutate this fast. So there's a good chance that what works this year would work next year too. So I think everybody's work is blessed, and we should bring as many tools into the market as possible.
So you're talking about an arsenal of tools to fight COVID-19, not one single track.
That's right.
I suppose
I never put all my eggs in one basket, so nobody should.
Good for you. Suppose the clinical studies start in 3 weeks. What is your expectation? When will you see the first results?
So we really hope to be able to see the first results in 2 to 3 months. That means, you know, I want to be able to say something conclusive before the next winter. You know, we're going to have a major problem, you know, coming September. The new flu season is up, and even in a regular flu season, hospitals are jam-packed. So the idea of passing another flu season with a COVID-19 epidemic on top of it is frightening.
Talking about flu, what is your take on Professor Yoram Lass's assumption that this is just another flu?
Well, it's not another flu, but for some reason, Israel has been significantly less hit than other countries. And you can see that in the numbers, at least in the first wave. We had 300 deaths in a country of 8 million people, while, you know, countries like Italy that have 10 times more people didn't have 10 times more deaths. They had 100 times more deaths. So Italy with 80 million people had close to 30,000 deaths, not 300. So something, well, you can call it divine providence, you can call it amazing clinical teams, and I think we probably have both. We might even have a younger population and more antibodies in the population for the new for other coronaviruses that are protecting us. But something is certainly different in Israel than other countries. So on one side, from the first wave, it seems like we have nothing to be scared of, but then I want to be ready just in case.
Talking about the flu still, might there be less flu this fall or winter season with fewer people gathering together?
We don't know. So what we do know is that there hasn't been a major breakout of flu epidemic in the southern hemisphere. That suggests that we might be heading into, mild flu season. Hopefully that's true.
Are you familiar with what's happening in the world, like Moderna, which just released the results in the New England Journal of Medicine, and the Oxford vaccine, which is currently undergoing final human trials with results promised to be published on Monday?
Again, I think those results are incredibly promising. One of the most incredible things that we noticed with COVID-19 is how fast the scientific and the academic world has been able to move forward with this. So let me give you a little bit of perspective. I was also working on hepatitis C and was part of the STAT-C program back in the day. So the virus was identified in 2000. It took several years until people could culture it. It took another year until we sequenced it. And then, the first drugs appeared, more than a decade after the virus was identified. Here, we realized we have a new virus in November. By December, it was cultured. Before January, it was sequenced. The first drugs, working or not, we're around, already in February with some results. We are talking about vaccines in July. That's 6 months. We've never seen anything move this fast. I know it feels like a lot of time has passed, but it has only been 6 months.
But most of the delays resulted from regulation, and now it's been easier than before.
I'm not certain. It takes time for things to move. It takes time for things to be sequenced, for cells to grow, for clones to be isolated. Even if the vaccine, let's say, is amazing, it works right now, scaling it up is going to be a process. The best estimates talk about 6 to 12 months. That means that we might miss the next. It might not be ready for the next winter, and that's a problem.
You mentioned side effects. Can you elaborate on that? What kind of side effects the medicine has?
For fenofibrate?
Side effects.
Yeah, so fenofibrate side effects usually happen upon prolonged use. So if people that take this as an anti-cholesterol drug take it for, chronically, just like statins. So you can have liver problems, you can have muscle pains. There are clear counterindications as well. Fibrates should not be taken if you have kidney dysfunction, liver or gallbladder disease, or any woman that is nursing. The advantages that we have here in this application is the fact that the new coronavirus infection is relatively fast. We can clear the virus from our system in about 2 weeks, without any external support. The biggest problem we have is controlling the symptoms. Okay, not the virus. The virus clears relatively fast. We need to control the symptoms. So taking a drug, whatever drug, shows the best clinical results, obviously, but taking a drug for 2 weeks, once you've been diagnosed with the virus or had early symptoms, and then, reducing the symptoms to the point where they don't bother you that much, the virus titer goes down so you're not as infectious, and then you will clear the virus anyway in about 2 weeks, is probably the best way to move forward.
You mentioned other drugs. So you tested all kind of drugs and then you decided the fenofibrate is the best? It's
certainly one of the best. We have other formulations we're looking at too. we tried to block glucose absorption into the cells, and there are a couple of drugs that showed some promising results, but they're not as good. We tried, PPR gamma agonists as well, so triglidozones. There's a bunch of drugs that we tried. Fenofibrate shows overall an overwhelming results. Even benzofibrate, we are not really certain that it would work the same way.
Suppose it works. I mean, the fenofibrate works. Is there a capacity to for companies to produce it in a massive scale?
So it's already produced in a massive scale. It's a generic drug. Everybody can make it. There are more than half a dozen companies that I think are already making it. Even Teva here in Israel.
I think Teva had a legal contest with the original producer, with Abbott Laboratory at the time.
Yeah, so it's a slightly different there are different formulations of it, but we can definitely give it to everybody that needs that. It's cheap, it's available, it's generic, it's off patent. There's a lot of things you can do.
Okay, sounds good to me, Professor Nahmias. Thank you for being with us, and we keep our finger crossed that it will work, and as soon as possible. I'd like to thank my colleagues at JPC, Talia Dekev and Rachel Exil, and Talia Aronsky, International Media Director at the Hebrew University of Jerusalem, for facilitating this briefing. Thank you all for being with us. Be safe. Stay tuned to our next webinars and have a great weekend. Thank you all. Bye-bye.